Welcome to the EvoEgg COVID-19 video series. In this video, we'll take a look at the innate and adaptive immune responses and discuss how they work together to eliminate pathogens from hosts and ultimately provide them with lasting immunity. By the end of this video, you should be able to describe the functions of 3 cell types of the innate immune system and 2 cell types of the adaptive immune system. The innate immune system is our body's first line of defense against invading pathogens. Immune cells, also known as white blood cells, are a variety of cells specialized for facilitating the immune response. Immune cells involved in the innate immune system account for around 3/4 of all immune cells. Since animals, such as humans, have co-evolved with viruses for millions of years, these cells have membrane proteins called pattern recognition receptors, or PRRs, that can detect various molecules which are common in pathogens, such as spike proteins. Generally, the innate immune system serves to keep the viral load at bay, providing time for the adaptive immune system to eliminate the virus and to provide the host with lasting immunity. Mast cells are found in large numbers in connective tissues and membranes. When a pathogen binds to a PRR, these cells release histamines, which in turn triggers blood vessels to expand, causing inflammation and signaling for other white blood cell types to come to that tissue and help deal with the virus. Although inflammation is often viewed as a harmful symptom of infection, it's actually part of our body's natural defense system against pathogens. For example, the dilation of blood vessels allows for white blood cells to be transported to the infected area quicker. It also results in an increase in local temperature and sometimes a systemic fever. Increasing body temperature can slow down the replication of the virus and, in some cases, may even kill it. Phagocytes are cells that envelope and destroy pathogens and dead host cells. They contain large lysosomes, organelles that contain enzymes that break apart cellular components that are no longer needed, or molecules that aren't supposed to be in the cell at all, like viruses. Macrophages are a specific type of phagocyte. They are large white blood cells that are particularly efficient at destroying viruses and bacteria. Dendritic cells are often the first white blood cells exposed to a new infection, as they are common in tissue types where viruses are most likely to enter the body, such as the respiratory tract, digestive tract, and skin. Dendritic cells bind to the virus, then engulf and transport the virus to lymph nodes, where B and T cells are abundant. It then presents these cells with an antigen by coupling them to a molecule called a major histocompatibility complex, or MHC for short. An antigen could be a piece of the pathogen, and in the case of SARS-CoV-2, the spike protein is used as an antigen. The presentation of this antigen activates the adaptive immune response. The adaptive immune response aims to fully destroy the pathogen, as well as provide the host with lasting immunity against the same pathogen in the future. Unlike the innate immune system, which responds in the same way to any invader, this component of the immune system functions by generating very specific responses that depends on the type of pathogen, hence the name adaptive immunity. These cells have specialized receptors that allow for adaptive responses based on the specific antigens that bind to them. When the T cell receptor binds to a spike protein antigen, it can differentiate into one of 2 types: helper T cells or killer T cells. Helper T cells are characterized by CD4+ membrane proteins, while killer T cells have CD8+ proteins. Helper T cells release specialized molecules called cytokines, which stimulate the activity of both innate immune cells and other adaptive immune cells, including killer T cells. Killer T cells, also known as cytotoxic T cells, destroy a target cell that is infected with a pathogen. It detects these cells by binding to the same antigens that originally stimulated its activation. B cells are primarily responsible for producing antibodies. Similar to T cells, there are 2 major types: plasma B cells and memory B cells. Plasma B cells circulate throughout the body and produce and release specific antibodies. These antibodies are designed in such a way so that they bind to the antigen that was originally detected by the B cell receptor. So in the case of SARS-CoV-2, they'll bind to the spike protein of circulating viruses, preventing them from infecting new cells. After the pathogen is no longer a risk, these specific antibodies are no longer produced by plasma cells. On the other hand, memory B cells create the same antibodies as plasma cells, but these antibodies remain attached to the cell membrane. These cells remain in blood long after the pathogen is no longer a risk. If the body is infected by the same pathogen again in the future, the pathogen is recognized by these antibodies, triggering the memory B cells to divide into plasma cells. This allows the host to have antibodies readily available to immediately deal with the same pathogen if it's infected again. This is why hosts may remain immune to infection from viruses they have previously been exposed to, and is also the primary mechanism that vaccines utilize to generate immunity. So, that's how the human body reacts in response to SARS-CoV-2 in order to protect us. In the next video, we'll take a look at how vaccines borrow from nature by generating an adaptive immune response without the high risk of disease. Well, that's all for this one, and we'll see you next time.